wild species have, in some Arctic environments, have evolved feathers on their feet to replace the scales. And we can see the same thing has occurred in domestic breeds of pigeons. Okay, different species of birds have evolved different beak morphologies adapted to different food sources. And we can see that breeders have selected for these different beak morphologies as well within the single species of domestic pigeons. And so you can even draw some comparisons between Darwin's finches with all of these radically different beak morphologies and what we like to call Darwin's pigeons, okay, taken out of variation in plants and animals under domestication, drawings of the different skull and beak morphologies of the different breeds of pigeons. And so we think that by understanding what's going on in this artificially selected species, we hope to ultimately be able to explain what might be occurring in wild species to generate some of the morphologies that we observe there. What's really nice, though, as I alluded to earlier, is that because these are members of the same species, it's possible to investigate them in ways that you can't investigate wild species. You can actually do genetic crosses. So this is taken from an old thesis from my alma mater, which is why I kind of stuck it in there. But it's showing the transmission of a particular phenotype from one parent to its offspring over time. You could never do this. You know, it would be fun to be able to cross a hummingbird to an ostrich to look at the evolution of body size, but you can't do that. But you can do similar things by crossing different breeds of pigeon. We can also capitalize on a lot of built-up knowledge over the years on domestic pigeon breeding, and also capitalize on a lot of the interest that's present even today in pigeons. So there are books written about pigeons. There are clubs of people who keep pigeons and show them as a hobby. So it's really nice, you know, for my day job, I spend time going around and visiting people that they work with the same organism I do as an escape from their day job. So I feel pretty fortunate to be able to do that. There are also pigeon shows where different pigeon breeders will come together, just like you've probably maybe seen the Purina Dog Show around Christmas time. There are pigeon shows as well, including one at the State Fairgrounds every November. And this is actually where we get a lot of our DNA samples from, is by collecting birds from these pigeon shows. And so we can capitalize upon all of these different resources. So I've spent some time introducing our organism. Let's talk a little bit more about why we chose to investigate color within this organism. One of the reasons why I think color is a really interesting characteristic is that it provides a good model for different processes during development. How are pigment-producing cells specified? How do they migrate to their final location in the embryo? How do they differentiate and produce the type of pigment that they end up producing? We also know quite a bit about pigmentation from work in other organisms, largely mouse and zebrafish. And one of the advantages we have is that because we're essentially trying to get pigeon up and off the ground as a molecular model, it sometimes pays to have stuff that's been identified in other species that we can kind of use a sanity check to see whether we're seeing similar things in our organism or not. This is also a genetically complex trait. In mouse, for instance, over 100 different genes have been shown to be involved in one way or another in generating color. And so this allows us to investigate how different mutations in different places of the genome might interact to generate the final phenotype. Also, this is an ecologically relevant trait. As many of you know, this is frequently the target of wild or sexual and natural selection in wild species, either for camouflage or mate choice or similar things. Also, it has relevance to you and I as well. So mutations in many of the same genes that we have identified in the pigeon have been associated with human either pigment variation or sometimes disease, such as albinism or predisposition to melanoma. All right, so I just want to take a brief moment to orient you a little bit and talk about how a feather ends up getting its final color. So the feather of a pigeon grows very similarly to the hair on the surface of us. And so it grows out of a follicle. And so at the base of the follicle are where the pigment-producing cells, their precursors, normally kind of reside. And so what happens is that as a feather is growing in, those precursor cells start to proliferate and actually migrate up and into the developing feather as they mature into melanocytes. So these are the pigment-producing cells here. And they actually secrete and deposit the pigment on the surface of the feather, so that by the time you're looking at an adult, fully mature feather, you're just looking at the end result, so to speak. The melanocytes have already done their job, they've died, and they're gone. But what is nice about this model is that if we want to, for instance, compare the levels of gene expression in the melanocytes from one colored bird to another color, all we have to do is pluck out a regenerated feather, and the cells are already there. We can harvest RNA from them and look at gene expression. So we don't have to do like skin biopsies or anything like that. It's just plucking the feather, which is really nice. Pigeons rely on the same type of coloring system that you and I do. They really only have two main types of pigment, two main types of paint, you can think of it. So one is a bluish, blackish color produced by the pigment eumelanin, or the pigment eumelanin has its color. The other main type of pigment is one that's called pheomelanin, that has this reddish, yellowish color. Now, both of these two pigments are produced from a common precursor substrate, tyrosine. And so the enzyme pyrosinase will convert that to another chemical called docoquinone that is either shunted into these two pathways depending upon the conditions within the cell. Now, spontaneously, docoquinone will end up being converted to pheomelanin without the action of any other enzymes. But under high levels of tyrosinase activity and coupled with other genes such as this one, it's DCT, we don't really need to talk about, but this one, tyrosinase-related protein 1, will generate the brownish blackish eumelanin. Okay? And so variations in the relative and absolute amounts of these two types of pigment are really what generate all of the different feather colors that we see in domestic. Now, we are actually not the first people to do genetics on pigeons. Okay, pigeons were actually used as a pretty good genetic model during the first half of the 19th century. So you can see here are three different papers, this one dating from 1918, these two dating from 1913, that have already looked at the genetic basis of color traits in pigeons. Now this, of course, is before we even knew that DNA was the hereditary material. So they're essentially just looking at patterns of how phenotypes are passed on from parents to offspring to understand the genetic basis. But it's actually quite incredible how much they were able to do just from inferring based upon outcomes of these processes. So for instance, let's introduce our first player in the color game, so to speak. So this is the major color locus that we're going to symbolize with a B. This is what a pigeon looks like if it's wild type at this major color locus. So you may recognize these guys, these are probably the most common color of pigeon that you'll see out in the city or whatnot. Now we've known through these classical genetic studies that there's a second allele at this same position in the genome called the ash-red allele that we're going to symbolize with this upper superscript A. Now you'll notice it has two main effects on the pigeon. It just turns a lot of the black feathers into red feathers, but it also turns the flight and the tail feathers sort of this ashy gray color, and so that's how it got the name ash-red. From these classical genetic studies, we know that there's yet a third allele at this position in the genome that we call the brown allele, which we symbolize with a lowercase b. And so it generates, instead of having a black or a reddish color, it has this nice brown color on the feathers. What's more, we know from these genetic studies, dating back almost 100 years now, that there's a dominance hierarchy among these different alleles, so that a single copy of the ash-red mutation is sufficient to have a bird produce the ash-red phenotype, whereas the brown mutation is recessive to both the blue and the ash-red allele. So birds need to have two copies of this mutation to have the brown phenotype. A second locus that we call the recessive red locus generates this nice rich red color over the surface of the entire pigeon. And then that is, I should point out, that occurs when the bird is homozygous for this mutation that we symbolize with the lowercase e. We actually know from these same genetic crosses that this locus is epistatic to this locus. Now what that means is that if a bird has two copies of this mutation, it's going to be this rich red color, regardless of what its genotype is at this position in the genome. So the mutations here override the effect of the mutations here. But up until this point in time, nobody had any idea why that was. And the last mutation, the last region of the genome I'd like to introduce is one that we call the loop. This is another recessive mutation whereby birds that have two copies of this have this lightened or washed out color. And so birds that are normally ash red, when they also have the blue mutation, are converted to this ash yellow color. Birds that are blue are converted to what the breeders call silver or dun. Brown is converted to khaki, and recessive red is converted to recessive yellow, so to speak. And again, all of this was determined just by crossing birds together and looking at the ratios of the phenotypes in the offspring. So what we wanted to do was to bring modern day genomic resources to bear on this problem, to see if we could actually identify the changes at the DNA level that are giving rise to these different phenotypes. So to do that, we drew upon data that we previously generated where we sequenced the genomes of each of these different birds here. So for the most part, it was one bird from each of these different breeds. The idea being that if we compare their genomes, we can start to try to identify regions that are responsible for different traits. So what I mean by that is that we take all of these red birds here, these birds whose names are in red, these are our ash red birds here. And we're going to compare their genomes to all of these birds in black, which are our blue birds. And what we want to see is, can we find any regions of the genome that seem to associate with the traits we're interested in? And so to illustrate that, okay, our first comparison was to try to identify the ash red mutation. So we're going to take lots of different blue birds, compare their genomes to lots of different red birds. The idea being that if we look at all the different sequences, we're going to find places where all of the birds are the same. Okay, so these are black areas that don't have any Latin mutant. So all of the birds are identical there because they're members of the same species. And so most of their genome is going to be identical. But we can start to find some places where certain birds have certain sequences that other birds don't. So over here, some of these have an A here, others have a C. And we see a similar thing over here. Some of the red birds have a C, others have an A. But ideally, we'll find some places in the genome where a bird's sequence will associate very highly with its phenotype. So in this example, all of these blue birds have a G here, whereas all of the red birds have a C. And so we would hone in on those areas as being the ones that are most likely to be able to explain the phenotype that we're interested in. And so when we did that, we found one gene that showed a significant association. So the sequence of the bird's genome in this gene explained whether it was ash red or whether it was blue. And it was in the gene that I mentioned earlier, TRP1, pyroxenase-related protein 1. And so what we're looking at here is a plot where each dot is just a different gene in the genome, and how high it is on the y-axis corresponds to its significance threshold. So a gene above the red line means that it shows a significant association by the phenotypes that we're grouping it by, whereas all these guys, they don't really show any sort of statistical association. So this was from our 40 birds that we sequenced the entire genome. Now it's not practical to sequence hundreds and hundreds of birds over the entire genome, but what we did was we took additional birds and we genotyped them directly at the TRP1 gene for the particular mutation we identified. And what we found was a perfect association where as every time a bird had one or two copies of what we thought was the ash red mutation, it in fact showed the ash red phenotype. And every time a bird didn't have a copy of this, it showed the blue phenotype. And because we knew from these classical studies that this was a dominant mutation, this result made a lot of sense. But what didn't make sense is the fact that we were picking up a gene that was encoding an enzyme that's supposed to be involved in one of the last steps in eumelanin synthesis. You'll notice that where it's located in the diagram here is well after the split between eumelanin and pheomelanin. And so we wanted to try to understand a little bit more about why it's causing this shift from eumelanin to pheomelanin when its normal enzymatic function is not thought to be anywhere near that pathway, part of the pathway. When we looked a little bit closer, we found that the mutation in TRP1 resulted in an alanine to proline change at amino acid number 23. Now the reason why that's significant, so here is a schematic of TRP1. It's composed of 7 different exons. And where it was at the very end of the signal peptide. So when the gene gets translated into a protein, the signal peptide is something that directs that protein to the ER, where it undergoes further modifications before being shipped back out to take place to help with melanin synthesis. And this was at the very end of the signal peptide. Now what I should point out is that the signal peptide is something that's normally cleaved off in the enzyme, in the ER. It's not something that's present in the functionally mature version of the protein. And when we looked at this position across multiple species, we found that every single vertebrate we looked at had an alanine at that position, just like our wild-type pigeon, and that the only time we saw a change was in our ash-red pigeon. So what this suggests is that over evolutionary time, it's been really important for this to be an alanine, okay? Evolution has selected for this particular amino acid until the ash-red pigeon came along. So that suggests that this mutation might be having an effect on the protein. The hypothesis we had was that this mutation might be inhibiting cleavage of the signal peptide. So normally this should be cut off. So what we wanted to do was to test whether that was in fact the case. And so we did a little bit of biochemistry. I'm not a biochemist, but I did the best I could. And so what I did was I took both versions of TERP1 and I put tags on either side of it. And I expressed these in cells. The idea being that this tag, this V5 tag, will show up both in the immature and the mature form of the protein, whereas the HA tag, on the other hand, should get clipped off as the immature protein gets converted into its mature state. And our hypothesis, again, was simply that this rate of conversion, the rate at which this HA tag and the signal peptide are clipped off, might be altered by the presence of this mutation in the protein. And so when I did the Western blot, I did find that that was in fact the case. That in both cases, we could readily detect the V5 tag that's present on both proteins, but with the wild-type protein, it was very difficult to detect the HA tag because it was efficiently clipped off and removed like it should be. But in the ash-red case, we could readily detect the presence of this tag on the protein. And so if we quantify that, we can see that the cleavage efficiency of the signal peptide was dramatically reduced by the presence of this mutation. And so this is causing the protein to not do something that, or not have something done to it, I should say, that it should be. So we wanted to know, okay, so the signal peptide doesn't get clipped off. How does that relate to what type of pigment the cell is going to produce? And so we sent some samples to a collaborator at the University of Cincinnati who did electron microscopy on the feathers. And what we're looking at here is a close-up zoom in on some melanocytes. Inside the melanocytes, the pigment's actually manufactured in little organelles called melanosomes. And they normally go through defined stages of development. So in normal feathers, you can see an immature melanosome is this nice oval structure with this nice lamellar matrix inside of it. And it eventually matures into this nice richly pigmented melanosome here. In the ash-red feathers, on the other hand, the immature melanosomes kind of look like a mess. They were circular and they have all these globular things inside of it. And the mature melanosomes don't have very much pigment at all. So that suggests that this mutation is somehow disrupting normal maturation of this organelle. Now, we stripped away the melanin from these feathers and we added DOPA, which is a substrate that the enzyme tyrosinase can work on. And we found some striking differences when we compared our feathers. So in blue feathers, or wild types, we could see that tyrosinase was active most highly at the edge of the melanosome, at the membrane here. Whereas in the ash-red feathers, the whole thing just sort of filled with the product of the DOPA reaction. So what we think is going on, and we don't have conclusive proof yet, but what we think is going on is that in the wild-type case, tyrosinase and TERP1, these two proteins, are sitting here at the membrane of the melanosome, and they convert tyrosine to eumelanin. But if that signal peptide isn't removed, it looks like this mutant protein is dragging tyrosinase away from the membrane where it should be, and that's somehow causing tyrosine to instead be converted to pheomelanin. And that might explain why this is a dominant trait. Okay? This protein is now doing something that it didn't used to do before. We next wanted to see if we could find the third allele at this position, which was pretty easy at this point since we'd identified TERP1. And so it's simply a matter of taking brown pigeons and sequencing the 7 exons to see if we could find any interesting mutations. By doing this, we found a nonsense mutation that some brown pigeons had in the very first exon. So this converted what should be an arginine codon to a premature stop codon. But not all of the brown pigeons had this mutation. So we sequenced some more. We found that other brown pigeons had an 8 base pair deletion in the second exon, and other brown pigeons had a 1 base pair deletion in the third exon. So what this actually means is that this trait is something that has evolved convergently in domestic pigeons. It's evolved multiple times. And if we count up the ratios of these, we can see that, while most birds have this mutation, most brown birds have this mutation, we can see that other numbers have the other mutations. And we even found one bird that was a heterozygote between two different mutations. So although this is a trait that has evolved independently in different lineages of pigeons, breeders must have been crossing them together over time, that in fact, both of these mutations have gotten brought together in the same bird, which is kind of neat. So to summarize this very first locus, we can see that different mutations are able to generate different phenotypes and show different bases of heredity. So this single amino acid change causes a dominant trait, the Ash-Red trait, to form, whereas these frame shift and nonsense mutations can generate this brown phenotype on a recessive basis. And so here's just a schematic showing how you can get these different colors just out of this single gene. So we next wanted to see what was going on at the next locus, the recessive red locus. And unfortunately, this time, our initial scans to try to identify regions of the genome responsible for this trait failed. And we started to wonder whether it could have to do with our point of reference. What do you guys notice about this bird here? Kind of hard to see, but does this look like a wild-type pigeon to you? No, not at all, okay? It has many, many kind of strange traits, and it's just sort of a historical accident that this was the one that they chose to generate the reference gene from. One of the interesting traits you'll notice that it has is it has this rich red coloration. Okay, which shouldn't be a problem if the phenotype is caused by just a single nucleotide change. But it can be a problem if there's a more dramatic change to the genome that results from it. And the reason is this. If this trait, or if this bird that is our reference genome has a deletion in it, when we compare all of our other genomes to it, where that deletion is, these sequences don't have anything to match. And those are going to be discarded as we go through our quality control pipeline. So that might cause us to miss something that we should see. So what we did instead was to turn to some gene expression analyses where we harvested feathers and looked at RNA expression. And we found that our B locus, TERP1, was highly downregulated in these recessive red feathers, as was a gene that's known to be an upstream regulator of TERP1, the gene SOX10. So this encodes a transcription factor whose job it is to turn on TERP1. And so this was really exciting, and we thought maybe we should look a little bit more closely at SOX10. So what we did was we took our pigeon genome and we lined it up against the genomes of other species that had already been sequenced, like chicken and zebrafinch. And what we found was that upstream of SOX10, there was about a 7,500 base pair region where there was no pigeon sequence in our reference that matched the chicken or the zebrafinch sequence. And so we went and looked at a bunch of birds, and we found that, yeah, not all pigeons have that deletion. It's only the recessive red birds that are homozygous for that deletion. But kind of similar to brown, we found that not all of the recessive red birds had this deletion. And so we looked a little bit closer and found that other recessive red birds had a different deletion that partially overlapped the first. What was really exciting was that when we looked at what's been determined in other species, we found mutant chickens and mutant mice that have deletions that partially overlap these that also show pigmentation defects. And if you notice, they all line up above this one highly conserved region, there we go, that has this asterisk here that's been shown to drive SOX10 expression in pigment cells. And so what we think is that this deletion, by removing this enhancer, is causing SOX10 to be turned off in recessive red pigeons. Now there's one way to try to prove that a little bit more closely, conclusively I should say, and that's by doing an allele-specific expression assay. So what we did was we got birds that were heterozygous for the recessive red mutation. Okay? So they had one copy of the normal gene, one copy of the mutant gene. Now we know that recessive red birds have a decrease in SOX10 expression, but there's different ways that that can happen. One way is that if the mutation isn't really all that close to SOX10, but it's in fact in another gene whose job it is to regulate SOX10. Now in this case, if this mutation is somewhere else, it should affect both of these copies equally, and we should detect levels of transcripts coming from both genes at about the same levels. On the other hand, if the mutation is in fact right next door to SOX10, if it's cis-acting in other words, what we should see is that in this cell, in this very same cell, this copy should be transcribed at a much higher level than this copy, because the mutation is right next to this one. So all we need to do is find places in the sequence where we can identify differences so that we know, which of these RNAs came from this copy and which came from this copy. And when we did that, we actually found that this in fact was the case, that the E2 mutation, this recessive red allele, was expressed at only about 10% of the levels of the wild type allele. And this is occurring in the same cell. Okay, this is all harvest from the same bird. And so that provides very strong evidence that the downregulation we see in SOX10 is due to a cis-actin mutation. This also allows us to explain why recessive red is epistatic to TRP1. When SOX10 is normally functioning, the bird's phenotype is going to be determined by its mutation, or what mutations it has at the TRP1 locus. But if SOX10 is turned off, TRP1 is going to be turned off. And that's why it doesn't matter what your genotype is at TRP1, the bird is going to have the recessive red phenotype. And so you'll notice that the bird in this case is red. It's not brown like you might expect based upon the other TRP1 null mutations. And so what we think is going on is that SOX10 might be controlling other pigmentation genes as well, and that when it's turned off, all of these genes being turned off is affecting the phenotype. All right, so we've identified 2 of the 3 main loci before, or so far, and so we wanted to see if we could identify the dilute mutation as well. And so we did very similar studies to what we did with TRP1, and we found one gene, SLC45A2, that showed a strong association with the dilute phenotype. And so just like with TRP1, we genotyped a lot more birds, and we found a very good association, whereas every time a bird was homozygous for the dilute mutation, or almost every time, there was one outlier, it showed the dilute phenotype. Whereas most of the times a bird was homozygous for the wild type allele, or heterozygous, it showed a normal phenotype. Now you notice there are a number of dilute birds in this category that don't have this mutation. What we think might be going on is there probably are additional places in the genome that can also cause a similar phenotype. And those classical breeding studies do suggest that is the case. Now not a lot is actually known about this gene other than that it affects pigmentation. So in humans with mutations in this gene, they show a form of albinism called oculocutaneous albinism type 4, I think. But so we do know that this protein is involved in pigmentation. And the mutation we identify in pigeon results in a histidine to arginine single amino acid change in one of the transmembrane proteins. And so what we think is going on is that this mutation might be affecting how well this protein can transport substances across the membrane that are involved in melanin synthesis. And that would probably explain the diluted washed out phenotype, which is caused by less melanin being deposited on the feathers. So we had these genomic association analyses, which was really nice, but it's also good to come up with independent lines of evidence to determine whether, to help support or refute your conclusions. So what we did was we set up a genetic cross between this guy, this handsome Pomeranian powder here, and this lovely female Scandaroon. And so you notice these two breeds vary in a lot of traits, but one of the traits in which they vary is their color. And so this guy has the ash red mutation. He also happens to be a carrier. He has one copy of the dilute mutation, but because it's recessive, you don't see it. The Scandaroon, on the other hand, she is homozygous for the recessive red mutation and the dilute mutation, so she has the recessive yellow color. And so when we look at their offspring, we can see lots and lots of different colors come out. And so what we can do is see whether the mutations we think are causing these traits, are these passed to the offspring that they should be based upon the offspring's phenotype. And so here are the different phenotypes we got from their offspring, and these are their genotypes down here. So we don't need to walk through every square, but the basic idea is that every wild type bird, or none of the wild type birds had a copy of our ash red mutation, or the recessive red mutation, or the dilute mutation. Whereas you notice these ash red and ash yellow birds, they always have the ash red mutation. All of the recessive red birds have the recessive red mutation, and all of the birds with the dilute phenotypes do in fact have the dilute mutation. So this provides independent evidence that the mutations we identified from the crossbreed analyses are likely the mutations that are causing these different traits. So in summary, we can see that all of these we've looked at 8 different color phenotypes in these domestic pigeons, and they can be explained by mutations of just 3 genes, TRP1, SOX10, and SLC45A2. And the way these different mutations interact in the organism is what ends up generating the ultimate phenotype. So we can get lots and lots of diversity from a relatively small number of genes, I think. And so we identified strong candidate mutations. It was interesting that in the course of this study, we identified both coding and regulatory mutations. We also saw that some of the phenotypes, like ash red, only evolved once, whereas recessive red and brown have evolved multiple times in domestic pigeons. This actually contrasts what's been shown in a lot of other domestic species, where usually one mutation explains all of the variation you see for a particular trait, such as body size in dogs. There's one main origin of small body size in all the different dog breeds out there. And as I mentioned, we can see that the combinatorial effect of just a few loci can generate a lot of diversity. And so I do want to point out that most of what I've talked about today came out of a paper that's hot off the presses in Current Biology last Thursday. So check it out if you're interested. But of course, as with any science, answers only lead to more questions. And so here are a few of the questions that we're starting to think about in the lab. One of the things I think is really puzzling is this ash red mutation. So you notice here are some wild-type birds. They have these black wing bars, black flight feathers, and black tails. But the ash red mutation, while it converts the wing bars to red, it actually converts the flight and tail feathers to an ash gray color. So why does this mutation affect different feathers differently? We don't really know. Also, we want to start to understand how color type and color pattern can interact. So here we have an example of birds that have the blue color, and they have the bar pattern on their wings. Here's a bird with the ash red color and what's called a check pattern on its wings. And we know from classical studies that this pattern is controlled by a different mutation than what's controlling the color. So in some cases, mutations will only affect one thing or the other, just the color or just the pattern. But in other cases, like with recessive red, this mutation actually affects both. It overrides not only whether it's going to be blue versus ash red, but it also overrides whether it's going to be bar and check, and it generates this uniform red color. And again, we have no idea why that is. Also, we do want to see if what we can learn from these domestic species does carry into wild species as well. So just like we have red and brown pigeons, you can go out in nature and find red and brown wild species as well. And so you might hypothesize that maybe mutations in TRP1 or differences in FOX10 regulation might be responsible for these phenotypes, where maybe other mutations in TRP1 or differences in TRP1 regulation might explain the brown phenotype of these offspring. So I'd like to thank you for hosting me. I'd like to thank everybody in the lab. So it wasn't just me that did all this work. There were a large number of us that contributed. Mike DeVos, Sydney, who's another grad student in the lab, another Mike, who's a lab tech, and Hannah, who's actually an undergraduate in the lab, who's done a lot of good work. And I know I just have a few minutes left, but I'd like to take questions from any of you that would be interested.
Available for questions. We'll need to vacate at about 10 minutes until another question What happens if you were to have another bird?
Looked at the interaction between diet and phenotype of the fish. That would be really interesting to see how nutrient levels might influence Yes, sir.
Just like over the class.
No, so interestingly, the TRP1 mutants, and actually all these mutants, they have these color phenotypes, but that's the only dramatic phenotypes we see. Now, what we don't know is whether, like if you look in populations living out in the city, whether there might be any selective advantages or disadvantages to any of these mutations. Most of the birds you see out there have the wild-type color, which would suggest that there are maybe some fitness consequences of these mutations that are subtle, too subtle to pick up by the human eye, but might show up in a more natural setting.
So, one more question is there any isozyme
be a heteromultimer, but I don't know that anybody, even in model organisms, has actually looked at the ratio of the two proteins in complex. They show that if you pull down one, you pull down the other. But I don't know whether they actually have looked that precisely to see how many of each molecule are associated with each other. That's a great question. Okay.
All right, we'll leave ended here. Let's give Dr. Domyan another round of applause.
Remember, 2 o'clock birthday cake.
Oh, yes, one other thing. Darwin's birthday cake will be cut at 2 o'clock today. Go up to the second floor, just up on the